My name is William Pomeranz. I'm an associate professor in the Department of Chemistry and a member of the Epigenetics Consortium. My lab is broadly interested in the molecular mechanisms associated with gene transcription. We use tools like structural biology, medicinal chemistry, biophysics to develop inhibitors of epigenetic complexes which can really control how genes are expressed. And so why we like epigenetics is it's really complex. We think about this in a simple sense, that if you think about a cell in its nascent stage, it has all the information it needs to be anything it wants to be when it grows up, but it ultimately has to decide when it does that in a tissue-dependent manner and a time-dependent manner. And how those genes are expressed over generations really underlies our interest in epigenetics. So what do we do? There are several proteins that are of interest to us in our lab. We're interested in nucleosome remodeling proteins, how these proteins move about the genetic information and how that affects both the fundamental biology as well as their roles in disease. So we study an understudied protein called BPTF. We develop inhibitors for these proteins. We developed the first inhibitor of a piece of one of these nucleosome remodeling proteins called a bromodomain, and our inhibitor was called AU1. And this molecule has allowed us to understand information about this protein beyond its function of simply binding to chromatin, but its role in both developmental biology as well as cancer biology. And so we're learning about this through a consortium of researchers across the country. We also study a protein among a class of proteins called BET bromodomains. And the challenge there is the binding sites are really similar, and developing selective inhibitors is really a challenge. In fact, molecules in the clinic have a lot of toxic side effects, and it obscures the biology that we can understand about them. So another thing that we've developed is molecule UMN 627, using our chemical biology tools, where we can now piece apart the function of a particular bromodomain called BRD4, which we're really excited about. And that's led to further collaborations with the Mayo Clinic, understanding its role in inflammatory liver disease. And so finally, what we do is we combine chemical biology tools, designing molecules that not only tell us about biology, but give emergent properties. Molecules that can degrade proteins, molecules that can bind multiple domains, synthetic transcription factors that localize our molecules to different regions in chromatin. And that, in a nutshell, is how we work in the field of what we call chemical epigenetics. So the work is important to me for the translational potential that epigenetic therapies can really have. This is a process where we can interrogate disease that's not being driven by simple mutations in DNA, but is dynamic and reversible. And there's so much basic science that we don't understand. So there's a lot of questions to ask on the fundamental level, but on the translational property, particularly in cancer biology, I think we have an opportunity to kind of move the needle by understanding some of these understudied epigenetic proteins. In my view, what the Epigenetics Consortium is it brings together researchers all across campus in disparate disciplines, where we've all kind of been doing our own research in epigenetics, but the field is so broad that we haven't communicated or connected with all the potential researchers out there, where we can have kind of a synergistic group of bringing to bear all our different specialty areas. And it's extended beyond the university to the Mayo Clinic as well. And so this is an opportunity to do bigger science beyond just the protein that we study in a test tube, but to translate our findings and to bring in experts from other areas. And so that's why I like being part of this consortium. So I was one of the early members of the Epigenetics Consortium. We take part at the most basic level through our collaborative research. We work with a variety of researchers, research labs, the Georg Lab, the Harkai Lab, the Trechnikova Lab in medicinal chemistry. We work with researchers in the medical school, and we work with researchers at the Mayo Clinic. And so our collaborative science is folded into this Epigenetics Consortium, using some of our chemical probe molecules to understand the biology. But more than that, our students take part in the Epigenetic Consortium seminars. We invite speakers in, not only internally, but externally, to bring in top-level science. And I help coordinate that as well by bringing in speakers. This has been incredibly useful to me to have consortium members that are experts in other areas of epigenetics to help my program. We work in a smaller area of research in structural biology and biophysics, and there are a lot more techniques that we can bring into our group. So we have experts in cell biology, we have experts in the cancer biology that we really need to help translate our science. We have experts at the Mayo Clinic which are thinking directly about translatable science to give our compounds new life as potential real-world therapies. So those resources are available to us, which makes this Epigenetics Consortium pretty special. The field has been working in epigenetics as a whole for decades, but now I feel like there's a nice watershed moment of bringing together a lot more chemical biologists and chemists and medicinal chemists to think about how we translate our basic fundamental science into real-world applications. There's excitement right now for epigenetic therapies, I would say, in the clinic. There are FDA-approved therapies for epigenetic regulated diseases, and I think those numbers are going to continue. And that's where I think the excitement is, once we can understand the fundamental role of a lot of these epigenetic proteins and the molecular mechanisms they're associated with, we have an opportunity for real change, I think, particularly in the disease biology. So the epigenetic therapy, that's where I'm most excited about. What makes working in the epigenetics field as Minnesota is there are a lot of people really excited about epigenetics. I'm not working in a silo. I have chemists interested in epigenetics. I have medicinal chemists interested in epigenetics. I have biochemists. I have the med school interested in epigenetics. There are a lot of researchers who we share a passion for epigenetics. And again, it's not siloed in just one department. So that's why I love being here at Minnesota, where there's tons of tools available to us, tons of expertise to actually do bigger science. There are a lot of opportunities, and we need a lot of specialty areas, specialty expertise that can help give us some new insight to these problems. There's a lot of room in the pool, so please join the Epigenetics Consortium. There are a lot of specialized techniques of just understanding kind of the big data that's available, understanding the chromatin level information, whether it's spatial organization, whether it's working with large RNA-seq databases, whether it's working with specialized techniques like ChIP-seq and identifying which particular proteins are localized at which particular sites on the genome. Chemists need to be involved of how we design these molecules to inhibit specifically these binding pockets over highly related epigenetic proteins. We need the chemists and the biophysicists and the biochemists to make the hard measurements in the test tube, and we need the cellular biologists to really look at the function at the gene level and the protein level in real physiological environments. So skill sets in cellular biology, molecular biology, structural biology, medicinal chemistry all can fold in. We need all those techniques to go after these problems.